Hello friends, welcome to my channel. Okay, today we're going to talk about the biology chapter, that is the first one chapter, that is reproduction in the lower and the higher plants. Okay, in this chapter we're going to talk about the reproduction system. So before we start, what is the reproduction? Reproduction क्या होता है हमें ये पहले जानना. So when you talk about the reproduction is a process in which organism continuity is maintained and the continuity of life as well as maintained, that we have to understand. And on the basis of the organisms, there are different methods of the organism to reproduce. On the basis of that, there are two types: asexual reproduction and the sexual reproduction, that we're going to talk about in detail. What are the types, on what are the subtypes of that, we're going to talk about this. As we discussed the definition of the reproduction is the production of the young ones like the parents. So there is a new generation, but it has a appearance or character like a parent. So that is a reproduction that we will talk about. That reproduction is very essential process for maintaining the continuity of the living organisms, because it's transferred gene from one generation to another and they are producing the new generation. For that purpose, second, it's maintain the continuity of life, that is more important. That is, reproduction is a, I can say, or we have to say that it is a basic characteristic of the living organism that maintains it alive. Reproduction, generation to generation are increasing. जनरेशन जैसे बढ़ते जाएंगे, किसकी वजह से बढ़ते जाएंगे? रिप्रोडक्शन की वजह से. सो दैट इज मेंटेन द कंटिन्यूटी ऑफ लाइफ. So these are the characteristics of the two of the reproduction. Okay, now there is each organism have their own method of the reproduction. On this basis, there are mainly two types. Broadly are distinguished into two types of reproduction. First method is that asexual method, that you all aware about this, and second one is that sexual reproduction method. So the underlying point or remarkable point over is that asexual method, which include only single parent, is required, but in sexual it required two parents, that is male and female has to be there. So why in asexual there is only single parents is required, but in sexual there are two parents. So there is a formation. The sexual is characteristic by the formation of the zygote, and that zygote formation required a fusion of the male gamete and female gamete. That's why there are two parents involved. But in asexual there are some process that we're going to study further, reproduction and the sexual reproduction. Talk about the detailed manner in asexual reproduction. Asexual reproduction, the first point is that there is no involvement of compatible gametes. What is compatible gametes? There is a fusion of the male gamete and female gamete which leads to the zygote, and the zygote has a, if it grow further, that is a different characteristic from the parent one. But in the case of asexual, that means not sexual reproduction, so there is no involvement of the compatible gamete. Here the word compatible means male and female gametes are not involved. See, as there is a generation or the formation of the new ones, young ones from the same parent, that is genetically identical progeny is there. What is the progeny? Progeny in simple manner उसको हम बोलते हैं offspring. It is a offspring. So genetically identical progeny, there are 2 points we have to consider. It is from the single parent. There is no other parent is involved, so it's from the single parent. So as there is a single parent, that means whatever the characteristic of that single parents are there, जो भी characteristic single parent के पास रहेगी वही transfer होगी. So that's why inheritance of a similar characteristics. So there is a two characteristic if the organism is performing asexual reproduction. जो भी पेरेंट्स की कैरेक्टरिस्टिक होगी वही ट्रांसफर होगी, एंड जो इनहेरिटेंस ट्रांसफर ऑफ द गैमेट इज देयर, इनहेरिटेंस ऑफ द सिमिलर कैरेक्टरिस्टिक्स. Now, as we mentioned here, only the single parent is involved. See, asexual reproduction characteristic involves only the single parent. So whatever is possess in the single parent, definitely those characteristics will resemble us to the young one. So same here, they are morphologically. What is morphologically? Morphology means what? The appearance wise they are same. They are identical by genetically. So there is a combination of the morphologically and identical. So whenever they produce asexually, the morphology and their identity are the same. So those are nothing but clones. Or this is also the process in which clones are formed. What is clones? Exact similar to the parents, that is called clones. Why? Because they are morphologically and identically same. So asexual method, we can say that produce the clones. Okay. Now see, in asexual reproduction, clones are formed basically, right? But there are many organisms which follow the asexual reproduction. There are subtypes. The first subtype is that fragmentation. What is that? Fragmentation, which is generally observed in the Spirogyra. We are going to talk about detail, but what is Spirogyra? If this is a Spirogyra structure, if I cut Spirogyra here, so there is a formation of two different Spirogyra. That's called fragmentation. Second submethod known as the budding system, and this budding is generally found in yeast. Okay, in this yeast cell, suppose this is your parent yeast cell, it form a bud. It form a bulge-like structure present on this that start expanding like this, and from the one parent only this bud is developing. If it get degenerated or separated from this, it form a new individual. So that's called budding, found in a yeast. And the third method is called spore formation, and this method is found in Chlamydomonas. Okay, it is found in Chlamydomonas. What is the beauty of this spore formation? From the one, suppose this is the Chlamydomonas structures. Okay, from this one Chlamydomonas structure, inside their gut there are formation of many new cells, and one that is break open these cells after maturity, there will be formation of much more chlamydomonatic cells. So what is the beauty or the this method is form a multiple new cell from the one parent cell. Here fragmentation, many forms by budding. Bud formation is the characteristic. Spore formation, multiple cells are formed inside the gut, and then whenever the cell break open, multiple chlamydomonas get released from that. So this is the beauty. Now we are going to talk about the each one in detail manner. What is the fragmentation? The word itself tell about that. Fragmentations means different parts में division हो जाना. It's called fragmentation. What happens exactly in fragmentations? See, basically it occurs in a multicellular organisms. The multicellular organisms break into the fragments, or why there is a word used fragments? Different parts, and those different parts are responsible for the generation of the new offspring. That's why it's called fragmentation. The example here is a Spirogyra. You can see image over the Spirogyra and the fragmentations. So if this is a Spirogyra cells, it break into the two parts, it leads to formation of the new Spirogaric two different cells. That's called fragmentation, as the name suggests. Now let's talk about the next type, that is a budding. The name itself suggests something. Budding, bud. There's something an outgrowth called as a bud. Now let's see here what happens in this. It is the most common in unicellular organisms. The earlier one, that's called fragmentation, which is occur in a multicellular. But now budding is the most commonly found in a unicellular organisms. We must know what is a unicellular organisms. The one cell which is responsible for all the cell activity, those are unicellular. So unicellular follow asexual method. With asexual method subtype is budding. In a favorable condition, there is a lead to formation of the one or two outgrowth. What is outgrowth? You can see in that image what is outgrowth is there. So 1, 2 outgrowth. That outgrowth are nothing but buds, and the name itself is what? Budding. So now separation of those buds, which further leads to the formation of the new individual. In this budding example, mostly common is yeast. You can see in the image as well shown in this video. There is a formation of the buds on the yeast. How it's formed? See, there is a one cell. How it's formed? There is a one cell has in a favorable condition. What is favorable condition? That is more important. फेवरेट कंडीशन का मतलब होता है सेल मैच्योरिटी लेवल आने के बाद रिप्रोडक्शन करना स्टार्ट कर देती है। तो पहले न्यूट्रिशन को गेन करेगी, यूटिलाइज करेगी, उसके बाद रिप्रोडक्शन हो जाएगा। सो नाउ when the cell, if there is a one outgrowth or two outgrowths are there, which leads to formation of what in the favorable condition only, and after separation of these birds, this leads to formation of the new individual cell. That is the beauty of this budding process. Now, what is the remarkable point? It happens in unicellular organisms. Outgrowths are called budding, and that's the process is called budding, and which is commonly example is what? Yeast. Spore and the flagellated, let's study about that. Okay, we are going to talk about the zoospore here. When you talk about the parent cell, and after the maturity, the chlamydomonas we are talking about, it will be form a new different cell inside the parent. But these cells are flagellated. What is flagella? If this is a tail and this is a tail-like structure is there, it's called flagella. So like they form a flagellated structure like this for the locomotion or the for motile. Flagella is very important. So that's why there is word used flagellated motile. So after break, this called a sporangium. Okay, why? It's a sac-like structure carrying all the zoospore. It's called sporangium. After getting break open, मतलब ये जब सेल ओपन हो जाएगा तभी this flagellated cells will come outside obviously, and these flagellated cells now are ready for grow independently, and this result of formation of the new individual. So now each motile cell now form different after growing. They form a new different individual cell. So in a chlamydomonas exclusively there is a formation, spore formation happens, and the spore formation leads to the multiple sporangium, that is flagellated motile zoospore. Why this is called zoospore? Because they are the tiny cells after maturity. What is maturity exactly? After achieving the proper nutrition, it undergo the growth and development and then reproduction. So again that is called zoospores. After the nutrition gain, they form a new big individual and then undergo the reproduction. So that's why there is word is used grow independently. Okay, so you must have understand this. There are three types others of the asexual reproduction are also listed in your textbook. So let's study about that in little bit detailed manner. You don't have to go in detail, but you must aware about it. What is it exactly? So other methods include first binary fusions in amoeba and the paramecium. Okay, in amoeba and paramecium. What is binary? When let's take example of amoeba only. So this is a amoeba. Okay, this is amoebic cell is there. See, after maturity, if it's cleavage into the two parts, so that one parent leads to the formation of new two daughter cells. So from one amoeba there is a formation of the two daughter cells. So that is binary. That binary indicates two numbers. So that's called binary fusion. Fusion means nothing but simply break down into the two parts. Okay, the next one is what? Chondria formation in penicillium. Acha, what is penicillium? Penicillium structure, if you look like that, it has a structure like this, having at the apex some little tiny particulate matter like spores. It is nothing but spores only, but in kind of different manner. You can see image in the video as well. Okay, now what is cornelia formation? Nothing but in penicillium. Suppose this is a penicillium. Okay, this is the structure known as a cornelias. Okay, that cornelia nothing but spores only. See, at the bottom it's a new, and the above, if you go above, it will become a little bit dry, but nothing but greenish it will be greenish in color only. So now what happens is that at the apex whenever the favorable condition happen, it fall onto the particular media and grow into the new penicillium formations, that is called conidia formation in a penicillium. Okay, you can see image as well which is put forward. Now the last one is the gamule formation which is found in the spores. Now when you talk about the gamule formation, what is gamule exactly? Is nothing but a propagule. What is propagule? Propagule is nothing but a branch which is having a having a nutrition food in itself. So whenever suppose this is any organisms is growing like this is a propagule. If break down into the two parts, the another branch will be formed like this. So now it act like a two new individual, that is called propagule. So what is propagation means what? आगे बढ़ते जाना, that is dule. Propagule having, it's nothing but a branch. Okay, in case of the sponges, image is put forward in your front of you, so you can understand that spores formation leads from the gamule formation. What is gamule formation here? The branch which is cut and that branch act like a new organisms or individual, that is called gamule formation. So these are the three things we have seen here, that is binary fusion, two parts form binary number, you have to understand, conidia formation in a penicillium, that is actually found in penicillium, and the last one gamule formation is a propagule formation is there. So we have to understand this. Okay, let's move on to the next topic, that is called vegetative reproduction. What is vegetative reproduction? See, plants reproduce asexually with their one of the part, that is called vegetative parts of that plant. So as they reproduce vegetatively parts, so definitely that part is belongs to the parent plant. So parent plant having those characteristics will transfer to the another plant, so same characteristic and transfer, so that's why they are genetically identical. As the new plant which is formed by the vegetative parts of propagation, they are genetically identical, definitely. So whatever the characteristic of the parent plants are there, so similar resemble to the next generation will be the having the same characteristics. So see, these are the in vegetative propagation, these are most of the time naturally reproduction not occur by these methods, but for the agriculture and the horticultural process for the human being as our requirement, we exploit the VR. VR stands for vegetative reproduction. See, if we have a fresh stock of the plants, we have to multiply it, so we need something to do with it. So that is the method found in agriculture and horticulture. So these are nothing but artificial methods which are not occurring naturally. For our purpose of the human requirement, we are doing so on because we want a new varieties of the plants, or the here written is that fresh stock of the plants we have to multiply it, so we follow some method. These methods are, first method is cutting, second one is tissue culture, and the last one is your cutting, tissue culture, and the last one is grafting. So what are the methods we are going to look upon each and everything in detailed manner. Cutting means simply means that we are cutting some parts and एक पार्ट कट कर रहे हैं उसको वही प्लांटेशन कर रहे हैं ओके सॉइल के अंदर, तो वो प्लांट आ जाएगा. फॉर एग्जांपल जास्मिन, इफ यू कट इट, इफ यू जस्ट एग्जांपल टेक एग्जांपल जास्मिन से अच्छा एक एग्जांपल हमारे पास रोज प्लांट, वी कट द रोज स्टेम. If you insert into the soil, give proper nutrition and the water, then definitely grow another new plant. So it's called cutting simply. Second is the tissue culture. In the tissue culture, we take a tissue from the parent in a nutritive media. We're going to take care of that inside the nutritive media. So whatever the callus form, it's called as callus. We're going to talk about detail in the biotechnology, not in this chapter. So in the tissue culture, we're going to talk about we get a tissue from the parent, we will grow it nutritive way, and then whatever the form, it has to be multiplied as tissue culture. And grafting, which is very interesting. In this case, we take a scion and a stalk. So we have insert two varieties of the different plants into the one variety. So what's that? We're going to talk about in a detailed manner. Okay, the first one method is that cutting. So here we're going to talk about the cutting. Cutting is the process in which it's the artificial method obviously. Small piece of the vegetative part, a small piece of any vegetative part, but what is the requirement? It should possess the buds. Definitely it should possess the buds. Buds होने चाहिए तभी हम लोग propagation कर पाएंगे. So what's that? A small piece of vegetative parts, that is the buds, is used for the propagation of this method. So any small piece of the any vegetative part of the plant can be a stem, leaf and the root. When you talk about the stem, it's called as the stem cutting, and the best example of is that rose and the bougainvillea. These are the example of this stem cutting, rose and the bougainvillea. You can see images over there. Now if the small piece having the buds, if it is a leaf, then definitely it's from the leaf the propagation can be occur. The example of this is the sansevieria. You can see image over here. And the last word, if the small piece is nothing but the root, then the propagation occurs. And the example is here is a blackberry. So what is important over here? The buds. Where the buds possess on the which part of the plant, that is more important, and that part is used for the propagation or the formation of new plants. That's why it's called a vegetative propagation and nothing but cutting, because artificially we are doing something on the plants. So that's it's called cutting. Cutting, three types are there: stem, leaf and the root, and these are the examples. The another method is we have to study is that grafting. The name itself suggests something, grafting. Okay, so what is grafting? There are two parts of the plant joined in such a way that whichever new plant is formed as the one single plant. There are two terms we have to understand in there: scion and stalk. What is that exactly? See, we have studied that two different plants parts are joined together, which will be act further as a one single plant. So there is a scion and the stalk is there, correct. Now when what is scion? The stem having more than one bud. Okay, scion is the one which has the desired characteristic having the more than single bud, and the stalk which is a rooted plant. Suppose this is your stalk because it has a rooted plant which is already exists there. So now what we are doing? We are doing a stem transfer which having a more than one bud to that on the scion. So if this is a insertion can be occur like this. If the insertion is like this is a stem that is called as a stalk having more than 2-3 buds. So we are transferring that stem having the more bud, that is termed as a scion, inserted on the rooted plant which is known as a stalk. So and this is bind together which is further act as a single plant. This called a grafting. We are going to talk about in detailed manner in the biotechnology chapter, which is very important over there. So right now we just must know that about that. The rooted plant is called stalk and the scion is the one having the desired characteristic and having one than more bud, जिसके पास एक से ज़्यादा बर्ड है जो प्रोपैगेशन कर सकता है, उसको हम उस प्लांट के पार्ट को हम लोग इंसर्ट करते हैं स्टॉक के ऊपर जो ऑलरेडी एग्जिस्ट है, रूटेड प्लांट है. So this is the grafting process. What is the beauty of the grafting added point? Grafting is very important. If suppose stalk is a one having a strong rooted system but do not possess any proper fruits or it do not possess any proper characteristic which we want. So what happens is that scion is act like a transfer of the desired characteristics. So here scion is a act like a desired characteristic. जो हमें न्यू कैरेक्टरिस्टिक चाहिए हम लोग सायन के द्वारा हम लोग स्टॉक के अंदर डाल सकते हैं. सो दिस इज द ब्यूटी ऑफ द ग्राफ्टिंग which going to talk about in detail in the biotechnology lesson. Here we should know about this only. Okay, now here one more term is that bud grafting. See, what is scion as a stem having the bud? If suppose I am transferring directly the bud, if I am cutting a bud from here and that bud is transferring to the another plant, it is also termed as a grafting only, and that is called a specially or exclusively called as a bud grafting. Bud grafting are apple, pear and rose as well. So this is the example of the bud grafting. The third one is a tissue culture. The name itself is that tissue. What is a tissue? You must aware about the tissue, plant tissue. Suppose we are doing propagation by using this plant tissue, then the process is called a tissue culture. Culture means what? Suppose I am taking a single part of the tissue, I have to grown into a particular plant, so definitely it has to go into the stages one by one. So till that time we are giving the nutrition, that is why it's called a tissue culture. So small amount of the plant tissue is taken from the parent cell, which is grown to, carefully grown to under a nutritive media, whatever the requirement is there, we are providing it, grown to into the plantlets. See, it directly don't form a plant, definitely the intermediate step with the plantlets, and these plantlets further grow and form your the plants. This is called a tissue culture. In this chapter they are not they haven't given a much detail in that. There is a one word written is that micro propagation. What is micro propagation? We know propagation means the plants को पढ़ने के लिए जो चाहिए, that is a propagation. Micro means for small parts. A tissue is nothing but a small part of the plant. So if using the micro propagation method, what is mean exactly? Suppose this is a XYZ plant which is the desired fresh stock is available, we want to multiply it. So if I'm taken any small cell or small part from it, cell from it, then if I undergo into the tissue culture, into the growing media, tissue culture into the growing media, it form into the callus. Okay, this box include means that there are some process undergone which is give the nutrition and all. The process it leads to the formation of the callus, and from that callus the new individual plants is growing. This is called a micropropagation. So tissue culture and a micropropagation goes in a similar way. Here we are using the any part. See, vegetative propagation means any part. We are, now we go into details in the tissue culture. When we talk about a tissue, that means what? Simply we are taking any tissue of that part plant and then we are growing by the micropropagation method or the tissue culture method. Ultimately our goal is to form the multiple plants over there, so that we are to achieve over here.